All right, so we'll begin. So the exam next week will be 50 questions,
So I'll start
multiple
chapter 2.
choice
So I'll be
on
recording
chapters 1 through
this part, and I'll post it like I did
there'll be multiple choice,
and
true
I'll
or
post it like I
false,
did chapter
and
1.
short answer, and we'll do a review at the end
So
of
if you
class.
so
if you look
talks
on
about
that
basic
if you
chemistry,
look on
the
that
building
file,
blocks
that have all the
of
course
living
materials,
things,
you'll see that there are these
atoms. So if you break a substance down to a
these
smallest
objectives.
smallest
So if you go
unit,
through
you'll get
if you go through
an
these,
atom. That is the
like
smallest
define
unit
an acid and a
of
base,
matter.
explain
And
the
those atoms
concept
are
of
made up of these
pH.
subatomic particles.
If you
Right?
go
We
through
kind of take
these
for
and answer
granted
these, you
that
should be able to
we've heard about these
do well
protons
on
and
the
neutrons since
exam.
we were
We'll
in
also do a
elementary school,
at the
but
end
it's amazing nonetheless.
end of the
Small,
lecture as well.
small units are what make us up as
So any
living
questions
things, what
on
make
the
up all matter.
any questions
So
on the exam?
protons, right? Those are these subatomic particles that have positive
if you go
charge.
on
Right? They make up those
Blackboard.
atoms. So this is a proton. Neutrons, they have no charge. And electrons, they have a negative charge. So protons are positive, neutrons are neutral, electrons are negative. But what's the big deal about these protons, neutrons, and electrons? So the whole reason that there's light within this room, that you're able to power your cell phone, is because these electrons, because electricity is the result of movement of charge. So this shows a battery. It has this side is negative, this side is positive, then you have a wire that connects the negative to the positive side.
The process
and that current is that movement of the electrical charge from one place to another. So those electrons are active within all of your electronic devices. So normally those
cancer is a result of when you have
atoms are neutral. They have the same amount of protons
and there are these certain
as
proteins called tumor suppressors,
same amount of protons as electrons. So you see this helium atom that has two protons, two electrons to cancel them out to make them
as well. So
to make them
class in
neutral.
chapter 2 we began
and then those electrons, those atoms, they hold their electrons within shells. So view them like a basket in which they can hold their electrons.
Which are
so this is a helium atom, and it just has two
positive neutrons.
It has two
neutral, and then those electrons have that negative
So there are
charge.
kind of three levels of shells in which you can hold your electrons. Within that first shell, it can hold
spinning around the center of that nucleus. And electrons
first shell
are
can hold
important because they facilitate electricity. So
second shell
electricity
can
is
hold
just the movement of charge from one place
and then the
to
third
another.
shell can hold
So if this
8.
this side is
if
negative,
this helium
this side
atom,
is positive,
if it had
then
more than one, if it had more than 2 electrons,
that electricity,
then it would
so
go
that
into that
charge is
second
zooming across
shell.
that
And we'll
wire.
talk about how you can have bonds within these electrons when they want to share. When they can have bonds between atoms when they want to share or donate electrons.
All those electronic devices are using that process. And then within your neurons,
so this shows some
your
important
neurons are like those electrical
elements within the human body. So oxygen that we need in order to breathe, carbon, which is the centerpiece, which is the building block of all living things. Hydrogen, which plays an important component of water. Nitrogen, which is an important component of protein. So all of these are important elements which make us as living things. And then calcium, we talked about how calcium plays an important role within bone density. Phosphorus plays an important role in bone density as well. Potassium plays an important role. Potassium,
defining characteristic of
fluid and electrolyte balance.
Of
iodine.
So raise your hand
to create, your thyroid hormones. All of these are important elements which make us up
an
as
atom. The number of protons. What's the name of the term?
What? So the atomic number of an atom is like the social security number of that atom. It refers to the number of protons. So atomic number is the number of protons. So think ANP. Think ANP. Atomic number of proteins. So atomic,
it has
not protein, proton. Atomic
all those atoms which have 2 protons
number
are considered
refers to the number of protons.
Helium. So
and that's the defining characteristic
have the same amount of
defining characteristic of an atom. So carbon has 6 protons.
They cancel each other
so
out.
all atoms which have 6 protons are considered to be carbon. So oxygen has 8 protons. So all atoms which have 8 protons
and then we
are
talked
considered
about
to be
how those different bonds,
oxygen. So that's the defining characteristic of those atoms. The atomic number is the number
ionic
of
bonds.
protons.
Those ionic bonds, they result from a transfer.
You can have a different substances
electrons.
Which can,
so those atoms, they hold their electrons within their shells.
A mixture of substances within fluid is called a solution. So the substance
and
that
the
is
first
doing
shell can hold.
the
Does anybody
dissolving
remember how many
is the
electrons
solvent.
the first shell can
Is the
hold?
solvent. The substance that's being
Second shell can
dissolved
hold
is the solute.
8. Third
So,
can
for
hold
example, lemonade.
8. Yeah, so
You have
that's that
lemonade
usual
powder, and
orientation
the
of the
lemonade
number
powder would be the
of
solute, and the
the
water
number
would be
of
the solvent. And within your bodies,
of those valence shells.
we
So
have
those
all these
valence
different
shells hold those
particles which
electrons. So
are being dissolved.
sodium,
so they would be solutes.
So this shows
And then
sodium.
the water within your body would be the
Sodium
solvent.
has atomic
So salt
number
water.
of 11.
The
So
salt
that
would be the
first
solute.
two within
The
that
water
first
would be
valence shell.
water would be the
put
solvent. So
those
make sure you know what a
next
solution
8
is. Like
within its second shell, and then it
combination of
has
substances
one
within
left over.
Fluid, the solute, that is what's being
And then we see
dissolved.
chloride.
The
It's gonna put
solvent,
its
that is
first
what's
two
doing the
in its first
doing
electron
the
shell, then its
dissolving.
second
So
8.
colloids, they
Then
have
it's gonna
larger
put
particles,
the
and
7
suspensions,
in the
where they
last
have
one.
very
So that
large particles.
chloride,
So, for example,
right, it's
it's
in
a mixture of
need
very large
of an
particles. For
electron
example, your
to fill
blood
its shell.
is an example
And then the sodium,
of
it can
suspension.
transfer its electron to
So this
chloride,
shows
so that one will be positive and one will be negative. So this shows
different
the process
atoms.
of that ionic bond forming.
This shows atoms which are inert. They aren't chemically active because they have their electron shells filled. So remember that 2-8-8 rule.
Sodium will be positive, and then just because one is positive,
The
one is
2-8-8 rule.
negative.
So this is
neon.
neon.
So those are weak.
It has the first shell can hold 2,
bonds.
second
So
shell can hold 8. So in the first shell it has
So that's
1, 2. Second shell it has
in water
8.
because water can
So it will have no need to want to react with anything else because its electron shells are
anybody
full.
remember, is water,
However,
polar or non-polar?
if your electron shells are not full,
right?
you will want to react with other atoms in order to fill your electron shells.
Positive
example, think if you have a
water,
basket
right?
of
The
lemons,
hydrogen
will your lemon basket can
the
hold
negative part of the
2.
chloride, and then the negative
I need to get one from somebody else. So I'll either borrow one from somebody else or share one with somebody else to get the amount of lemons in my lemon basket. So this shows that helium. So that first electron shell can hold how many? 2, and this only has 1, so it's very reactive. And this shows carbon. So its atomic number is 6, and its first shell it is full because it has 2. But its second shell, the second shell can hold how many? 8, and it only has 1, 2, 3, 4. So it can react with 4 other structures. So that's why carbon is such an important atom, such an important element within living things, because it can react with 4 other structures. It's like your body. Right? You have 4 limbs which can grab onto other structures. So carbon, it can bond with other structures. So that's what makes it
rip apart those
unique and so special.
And then we have covalent bonds. So this forms the oxygen molecule.
So you can have bonds which can occur
a sharing
when those atoms they either want to transfer one electron to another atom, or if they share electrons. So for example,
we see oxygen. So that first shell can hold.
We'll look at sodium. So within sodium's first shell, it has 2.
Right, second shell can hold.
So it's
8, but it only has 6 within its outer shell.
So in sodium's second shell, it has 1, 2, 3, 4, 5, 6, 7, 8.
2 in its first shell,
second shell is that full?
6 in its outer shell. So that
but in its last shell,
we're going to pair up.
It only has 1.
To one another.
So it needs 7 other to be full.
Then they form these.
And then now we'll look at chloride.
Bonds.
So chloride in its first shell, it has
sharing valence
2
electrons.
In its second shell, it has 1, 2, 3, 4, 5, 6, 7, 8.
Oxygen forming, right?
It's full, but in its last shell, it has 1, 2, 3, 4, 5, 6, 7.
And become.
So all it needs is 1 electron to be full.
And ionic bonds.
Right, so sodium says to chloride, okay, I'm going to do you a favor. I'm going to donate my electron to you so you can be
polar and non-polar. Right, so these polar covalent
what sodium does, it transfers its electron to chloride, so that chloride will have a full electron shell. So when you, let's say you're in a group of people,
these non-polar
covalent bonds, gain a negative person, as your group become more negative.
So you see those with lipids. You see those
they become more
fats.
negative.
They are.
But if your group loses a negative person, you become more
equal share
positive.
Of electrons.
Right? So look at sodium.
So
Since it lost the negative charge, it became more positive. So
water and oil don't mix.
And since chloride, since it's gained a negative charge, it becomes negative. And because we know the opposites attract, right? They are attracted to each other because one is more positive and then we talked
about hydrogen bonds,
ionic bonds. That's an example
water
of an ionic bond.
Forms this network
of a transfer of electrons. So ionic bonds form as a result of a transfer of one electron. Sodium from one water molecule will bond to the chloride.
Hydrogen, right? The positive
ions, those are kind of separate dissolved ions which have a charge. So ions have charges on them. So ions, those are structures which have charges on them. So this is sodium ion
because it has these hydrogen bonds which help to connect.
Chloride ion which has a negative charge. So those ions, they have charges.
And then we talked about how you can have synthesis
so any questions on
what's an example
ionic
of
bonds and how they form?
Of a synthesis reaction in the body. Anyone could think of one?
They result as a transfer of electrons. So sodium, like when you transfer this electron,
when you build the structures which make up your body, when you're building proteins within your body, you're creating something. When you build fat within your body, you're creating something. So
chloride becomes more
that's
negative,
something small,
sodium becomes more positive,
to build up something large.
Opposites attract,
small components
to hover around each other.
A large component.
So those are kind of
amino acids. So
those are weak bonds.
Before you go to the gym, you drink
so
a protein shake.
They're not married.
And that protein gets dissolved into its amino acids.
Marriages that occur,
then you want to build a protein within your own body. So
atoms, those are called
amino acids that you consumed.
They're called covalent bonds. Remember, those shells are called valent shells.
Those amino acids
so
to create that
cooperate with somebody else. You're working with that other person.
Taking those small amino acids,
they share building blocks. So
to build up that large structure
how methane is formed.
A protein, and then
so you got
decomposition reaction. That's when you break down something large.
So that first shell can hold
small. So this just shows glycogen. Glycogen is outer shell can hold. It's a storage,
but it only has 4
of carbohydrates within our body.
To have 4 other electrons.
You've gone a long time without eating.
This hydrogen,
your body is not going to let you.
It only has
starve.
1. Remember that first shell, it can hold.
Right. Remember that homeostasis.
2. So it only has 1.
You have the storage tank of glycogen, of carbohydrates. It's within our liver.
These 4 hydrogen ions,
and within our muscle.
Right? We need 1 to be full,
your heart,
and then it sees carbon. And carbon needs 4 to be full. So they'll say, why don't we work together? Why don't we cooperate with each other and work together? So this helium will bond with this
you've gone a long time without eating.
We'll share electrons with this with this
your blood sugar
part
drops.
Of the carbon. Then this helium
atom
will share electrons here. And then this hydrogen ion will share electrons.
Is that positive or negative feedback?
So they'll all be sharing
feedback
electrons.
Because it causes an opposite
so
response all be full. To that initial stimulus.
So now, so if you look here,
now
this hydrogen ion, it has 2
so
within the shell. Now this hydrogen ion, it has 2.
Release a hormone called
this hydrogen ion, it has
glucagon.
2. This one has 2. And then this carbon, it has 1, 2, 3, 4, 5, 6, 7, 8 within its outer shell. So by cooperating with each other, they link together and share electrons. So covalent bonds, they result from the sharing of electrons. So covalent bonds result from the sharing of those electrons. So make sure you know
it's going to stimulate the breakdown of
and
glycogen
covalent
into glucose.
Bonds. Covalent bonds result of the sharing of electrons,
which will increase the blood sugar.
Result from
so
the
when
transfer
somebody
of electrons.
is diabetic, let's say they took too much
so
insulin and they got a
you can
big
also
drop in their
have
blood sugar,
an unequal or equal share of electrons within covalent bonds. So for example,
the blood sugar drops into very dangerous levels.
This
shows
give them
a shot of glucagon to pump up their blood sugar.
This shows how you have kind of an equal share of
electrons
electrons
into glucose. So
carbon
that's an example
and oxygen. So when you have an equal share of electrons,
that large,
it's called non-polar.
Unit of carbohydrate,
that glycogen. So a pole.
See how many
that is when you have
together, right? All these different links,
2 different charges on 2 different areas. So you look at a battery.
Blood sugar, and then when you go into the gym,
battery is polar. One side is positive. One side
body
is negative.
breaking down that glycogen.
Polar. Right, but if you have
within your muscle
an equal share of electrons, right, there's no polarity to it. You have an
glucose to
there's
fuel
an equal distribution of the charge within that structure.
The contractions of your muscle.
A non-polar substance. Water, on the other hand, it is polar.
When you burn fat,
that oxygen hogs all the electrons.
The adipose tissue,
so oxygen is negative
into those fatty acids,
and the hydrogen is positive.
You can use to fuel the contraction of your muscles.
So this shows that H2O.
So that's an example of a decomposition reaction.
Oxygen negative.
So make sure the difference between synthesis and decomposition, and then exchange reactions.
So
there's
just
this
like
energy
that battery had
our bodies
a positive side and a negative side.
The battery
this
of the
water
body
has a negative side and a positive side. So therefore, it is polar.
And it will transfer
unequal share of
high energy phosphate
one side is positive,
onto different structures.
One
to help
side
to
is negative.
To energize.
And because water is polar, it is what gives it all its special characteristics. Gives its characteristics. That's why water is a good solvent. That's why water is able to dissolve different substances.
Transferring this high energy
them apart based on their charge.
I kind of view ATP
to make sure
as
that water
like,
it is polar. Water is polar. One part is negative, one part is positive.
ATP is kind of like, ever see the movie Fantasia where Mickey Mouse like brings life into that
so this describes
so that's what
hydrogen bond. So we know that
ATP
the
is
oxygen part of
these different
water
structures and
so
create
oxygen part is it negative or positive?
Create movement within them.
negative. So the oxygen part is negative, and the
And then
hydrogen
acids
it is
and bases. So we
positive.
talked about the
So
pH scale. So if something is
in water,
below
it will form bonds
7,
between
is
each
it
other
acidic
like
or
a
basic?
group of skydivers linking
Acidic.
hands
Above
together.
7, it is.
So the
Base,
negative
yes, it is
of
basic
oxygen
or alkaline.
will
So
bond onto the
A,
positive
A
hydrogen
comes before B in the
of
pH
a
scale
neighboring water molecule.
in the alphabet.
And then the
And
positive hydrogen will bond onto the neighboring negative oxygen of another water molecule.
Bases in the pH
So
scale,
they'll
just
form
like A comes
bonds
before
between
B
each other.
in the
That's called
alphabet.
hydrogen
So
bonds.
go from 0
Those are bonds that occur
to 14.
between water molecules, and that's what's responsible for all the unique characteristics of
7
water. Why water has a high boiling point,
So 7
because it has these
neutral.
bonds which occur.
So what makes something an acid? What makes something a base? So if something donates a hydrogen ion,
so
it is considered to be
chemical
acidic.
Chemical reactions that result from either breaking apart bonds or the building together of different types of bonds. So these reactions are constantly occurring within your,
which we have within our stomach,
occurring within your body. So
if
a
you put it in a
synthesis
water
reaction,
solution,
think you are creating something. You take these small units to create something large. So
hydrogen.
The building blocks of proteins are amino acids. So let's say you eat a steak. You break it apart into its amino acids.
Get rid of that hydrogen.
And then you take those amino acids to build the structures within your
body,
right? So to build
can
muscle,
monitor
right?
the acid and base content of the body. There's these structures called chemoreceptors, which monitor whether there's a lot of hydrogen ions and
decomposition reactions. Those are when you break down something large.
Your body works to maintain constant.
So, for example, in your liver and your
we like to maintain
in your liver and your muscle,
7.7.
There's a storage unit of sugar called glycogen that your body will break down when it needs sugar. So this shows the liver.
Like 7.35 to 7.5. Kind of maintain that around 7.35 to 7.5. So this says goes around 7.4.
So when your blood sugar drops, your body will respond by trying to bring it back.
If it gets too acidic, like carbon dioxide acts like acid within the body. So these chemoreceptors, they'll monitor if there's a lot of hydrogen ions
in the blood sugar. Your body will break down this storage unit of carbohydrates, break down this glycogen,
carbon dioxide, which is an acid, or will urinate out hydrogen ions to bring that pH level, back up. So pH is a very important.
Break it down to glucose to bring the sugar back up.
Constantly,
so it does it by breaking apart these little linkages of glycogen to break them down into a glucose. So that's an example of a decomposition reaction. Break down something large into something
but it also serves a defensive function as well, because
and exchange reactions occur when you exchange different components of different molecules.
Maintain it as acidic.
So this shows an energy unit of the body called ATP.
You don't have to know it, but
it will transfer this P, this phosphate group, onto glucose in order to
carbohydrates.
So it is kind of giving its component to that
lactic acid.
Other molecule. So synthesis, break down something,
which helps to maintain that pH. So if you take
synthesis, you take something small to build up something large.
Sometimes if you kill the good bacteria,
sometimes if you kill the good bacteria, which can lead to exercise and your muscle grows. That's a synthesis reaction. Building something
the vaginal pH, it can lead to different
decomposition. You take something large and break it down into something small.
Serves an important glycogen into glucose to pump up your blood sugar. Body, and then those bases they accept hydrogen.
So substances can react in unique ways with
a hydroxide ion, which is
substances
acidic.
That release hydrogen ions in water are
something
acids.
negative will attract something positive. So those bases are usually negative, and they will attract those positive hydrogen ions to them. So make sure you know
acid.
So if you saw an example of a pH of 7, it is.
So for example, hydrochloric acid, 5, HCl.
Make sure you can say that it is acidic. So if you saw those pH values, be sure that you'd be able to recognize them.
Within water, in that H2O.
So this shows how we are able to create different molecules.
Oxygen is negative, hydrogen is positive.
Macromolecules, the building blocks which make us up as living things, right? The carbohydrates, the
hydrogen
lipids,
will be attracted
the proteins, which we
oxygen
consume and which help to build the structures within us. So
there
are
ways will be attracted to the hydrogen. So dehydration will rip it apart, and you will release hydrogen ions.
That's where you're able to create a structure
hydrogen
by
ions are
taking water from them. So
and structures that release
organic molecules,
ions called hydroxide.
They have many units which make them
hydroxide.
Up, called monomers. So monomers,
they're called bases,
will combine to form
bases. They
polymers.
accept
many units of monomers combine to form polymers. So if you look at
they release those hydroxide ions. And bases, they also accept hydrogen. Bases accept hydrogen. So hydrogen is positive.
Many units of an
individual. So what would attract something that is positive?
Many units of
something that is negative. Unit. So bases are usually negative.
These larger structures
they're able to bind and attract
this monomer has
those
OH,
hydrogen ions,
and this one has an
and so
H.
so make sure you know that acids
H2O,
release
that is
hydrogen. Acids
water.
release hydrogen, and
so if you
usually positive.
take away water,
hydrogen acid. Bases are usually
they will
negative
link together,
and they accept
and
hydrogen
and form a unit.
So make sure you know that. That's very important.
Monomer.
The pH scale,
taking away water,
that measures the amount of hydrogen within a solution. So pH means the power
a monomer.
So hydrogen.
If this was a
and
carbohydrate,
the pH scale goes from 0 to 14.
You have these 2 units.
I think it goes alphabetically.
And then you're able to break things apart. So this is why water is so important.
So just like A comes before B in the alphabet,
it helps to break down different structures.
Acids come before bases in the pH scale.
Meaning water, lysis meaning the breaking apart. So this shows.
So 7 is
monomers which are linked together.
So
if you
below
add
7
water,
is acidic.
Split it apart.
Below 7,
so hydrolysis. By adding water, you
acidic.
will break those bonds and split them apart. So
above 7,
so by adding water,
is
will break them apart.
So think
by adding water,
before B in the
alphabet,
acids come before bases.
Counterintuitive.
Acids come before bases.
By taking away water, you bring things together.
So something that has a pH
between them. So think
5. Is it acidic or basic?
Then if you take it away,
pH of 10, it is
both come together,
basic.
And then you have
so this
bonds between those
shows an example.
If you add water,
some common acids and bases. So
ammonia,
that is a base. Bleach is
an example. Your blood
is a little bit basic.
An example of a monosaccharide. This is just one unit of
another
word
for basic is alkaline.
And
so alkaline and basic.
Another example of a monosaccharide. One
example
unit
of acid. So lemon juice and your stomach has that hydrochloric acid
OH,
because it's very low pH. So pH balance. So H is something your body is constantly working to maintain
water. So if you
so your skin has a certain pH, so it won't allow the growth of bacteria. Your stomach has acid within it to break down different structures, but also to kill different bacteria, right? Even the vaginal pH is acidic, so that certain other organisms won't be able to grow. So we need to maintain this constant pH balance.
So next time you put some table sugar
within the body.
I know, there's strawberry or whatever, right? And you drink water. Think, right? That water is breaking apart that sucrose into glucose and fructose. So that's a hydrolysis reaction. So now we'll talk about those major organic molecules, the carbohydrates, the lipids, and the protein. They're called organic because they have carbon. So they're called organic because they have carbon. Carbon is like a linchpin within all living matter. So within all those organic compounds, you'll find carbon. So I'll talk about the different monosaccharides, disaccharides, and polysaccharides, and make sure on the exam I give an example of a monosaccharide, of a disaccharide, of a polysaccharide. So carbohydrates, their major function is fuel. They're the main fuel source of the body. They're the main fuel source of the body. And then glucose, this is our fuel, right? So that glucose is an example of a monosaccharide, and it is the fuel for humans and fuel for living things. So make sure you know glucose is a monosaccharide. Because in lab, we talked about that process of cellular respiration. Right? That's the reaction of life. We take the foods that we eat, we break them down into glucose. Let's go back. Combine it with oxygen from the air that we breathe. So right here. So this shows that reaction of cellular respiration. You take glucose from the food that we eat, so that C6H12O6, right? That glucose, full of that carbon. Combine it with oxygen from the air that we breathe to produce that ATP, that energy molecule of the body. And as a byproduct, you produce carbon dioxide that you exhale, and also water. So I make an analogy between our cells doing that cellular respiration and the combustion reaction within a car. So the cars, they need that carbon source in the form of octane, combine it with oxygen, right? They can create energy, and they release that carbon monoxide and carbon dioxide. And then we use that ATP to fuel and power the firing of our neurons. It facilitates the contraction of our muscles. Every movement within our body is powered by that ATP, just like the gas powers your car. Very important reaction within the body. So another example of a monosaccharide is fructose. So if you eat an apple, right, the sugar that you have within that is fructose, right? We find fructose within our fruits. So we have Within fruits, we have fiber, which is like the good stuff, and then we have the fructose, which makes it taste good. So the fructose makes it taste good. So they're able to produce those carbohydrates through photosynthesis. So light hits those plants that you see outside. So light, and then carbon dioxide from the air that we breathe. So they take the light, they take that carbon dioxide to produce the carbohydrates, which make them up. So when you eat an apple, you're eating somebody's breath that is combined with light that has come to life. So that fructose, we get that fructose from the fruits that we eat. Another type of monosaccharide is galactose. Another type of monosaccharide is galactose as well. And then, so we'll talk about DNA. Our DNA, it is deoxyribonucleic acid. So our nucleic acids, a component of them are sugars. Sugars make a component of our nucleic acids. The very recipe, the very instruction manual, which makes you up as a living thing. So this deoxyribose, right, this deoxyribose plays a role in making our DNA, your instruction manual. And then this ribose plays a role in making RNA, which is another form of genetic information that the DNA turns into, and we'll talk about that. So some examples of disaccharides, we talked about that table sugar, that sucrose, which you will dip in your strawberry, drink in water, and then think that there's a hydrolysis reaction going on within your body. Right? Maltose, and then lactose. Lactose is an important disaccharide that we find within milk. So so normally within our small intestines, there's an enzyme called lactase. That enzyme, it helps to break down that so let's say this is lactose. And within our small intestines, there's an enzyme called lactase. That lactase will break that. So that lactase is an enzyme that helps to break down that lactose into glucose and galactose. So that lactose will go in, and out will come these monosaccharides. But when people are lactose intolerant, they're deficient in that enzyme lactase. So they're not able to properly break down and digest that lactose that they find within milk. So this is lactase. So when people are lactose intolerant, they don't have that lactase. They have that buildup of the lactose. So if they don't digest the lactose, the bacteria within their digestive tract will, and they'll consume it, and they'll produce gas as a waste product, which cause some of the different symptoms of lactose intolerant. And also water follows solute. So water will follow solute. So water will follow that lactose into the intestines, and it will cause diarrhea. It will cause diarrhea as well. So that's an example of an application of that knowledge of what a disaccharide is. And then, so polysaccharides, some examples of polysaccharides are glycogen, right, that storage unit of carbohydrate that we find within our liver and our muscle. So starch, right, starch that you find within your pasta, right, is an example of you find within our pasta, that you find within your rice, you find within your fruits. And also, there's another polysaccharide called cellulose that you find within plants. So cellulose, that's an indigestible carbohydrate within plants that makes up fiber. So that's cellulose that makes up fiber. So your body is not able to make that, it's not able to digest that cellulose. It's not able to digest that fiber. So it helps to kind of push the contents of your food along your digestive tract, along your large long intestine. So if people have a constipation, they suggest giving them a high fiber diet because it will help push the food along their digestive tract. And also, since your body doesn't digest it, your body kind of burns calories when it's trying to digest that fiber. And then so when you're trying to digest it, you're moving around, you're walking around, and that kind of burns calories as well, because as you're trying to digest it, it kind of takes, your body is moving, and that burns calories as well. So those monosaccharides, like for example that fructose, get digested very quickly. They get digested very quickly. But those complex carbohydrates, they take a while to digest. So make sure you know that glycogen is an example of glycogen is an example of a polysaccharide within your liver and your muscle as well. So lipids, so fats, they're called triglycerides. So you have different types of lipids, so different types of lipids. So one type of lipid that we find within our body are these triglycerides. So that's that storage form of lipids within our body, triglycerides. They're called triglycerides because they're composed of a glycerol molecule. This is a glycerol. Then they have these 3 chains of fatty acids, right? These are called fatty acid chains, so right here. So it's like somebody has a head and they have 3 strands of hair. So this is the glycerol head. Then they have 3 strands of hair within the fatty acid chain. So these are the fatty acid chains. So I'll show a video which will describe cholesterol, which is an important lipid within the body, and also the different roles of lipids within our diet. All right, so cholesterol, that's the building block for what type of hormone? Which type of hormones? Testosterone and estrogen. Very good. Testosterone and estrogen. And then so the cell membrane is made up of what type of lipid? Phospholipid, yeah. So make sure you know those triglycerides, those are how we store fats within the body. Like cholesterol, the building component of steroids. Make sure you know cholesterol. Cholesterol forms what substance that's produced in the liver, stored in the gallbladder, which helps to break down fat. Bile, very good. Helps to create bile. It helps to make sure you know those lipid soluble vitamins. So the function of vitamin D, anybody? If you raise your hand. Anybody? Go ahead. Calcium absorption. What are some functions of vitamin A? Anybody? Go ahead. Vision, cell division. Vitamin K, clotting, and E is in a antioxidant. So I'll run through the rope real quick. Samantha, Michelle, Kristen, Cheyenne. How do you pronounce that? Cheyenne, Sean, Vanessa, Ruth, Deanna, and Ephania. Did I say that correctly? Stephania, Chad, Ashley Mendez, Jessica, Jennifer, Rochelle, Dylan, Shanokinen. How do you pronounce that? Shanokinen, Jeanette, and Jessica. You're not on that. So you think maybe, oh, Jamaica Stevenson. So lipids also form the basis for hormones within your adrenal glands. So adrenocortical. So your adrenal gland. So you have your adrenal gland right here. I mean, so you have your kidney right here. And at the top of your kidney, you have the adrenal gland. And then the inside of your adrenal gland is the medulla, but the outside is called the cortex. So it's like a Tootsie Roll pop. The outside is that hard candy, inner part is the Tootsie Roll. So the outer part, the hard candy part of your adrenal gland is called the adrenal cortex. So you have hormones which are released from your adrenal cortex, which are produced from lipids. So cortisol, right? Cortisol, that's an important hormone which plays an important role in the and it's an anti-inflammatory. So you ever put like cortisone on the surface of maybe a bug bite, or you took some corticosteroids if you had some inflammatory disease. So corticoids, they play an important role in decreasing the inflammatory response, and they can stimulate the breakdown of protein. They stimulate the breakdown of protein to create glucose. So the breakdown of protein to create glucose. Then aldosterone. Aldosterone. So when you hear that "on," it's referring to like a steroid hormone or a lipid hormone. So aldosterone, it plays an important role in water regulation. So it will stimulate, so this shows the kidney tubes. So next to your kidney tubes, you have blood vessels. So we talked about how the last class, how the kidney plays an important role in water balance. So these are the filters within your kidneys. So blood will go in. This shows a blood vessel. Will go into the tubes and out will come urine. So this U is urine. So aldosterone, it will stimulate. So if you get a drop in blood pressure, it'll cause kind of a chain of events which will cause the release of aldosterone from your adrenal glands. It's aldosterone, and it will stimulate water to go. It will stimulate, I'm sorry, sodium to go from your kidney tubes to your blood. So it causes sodium, this Na plus is sodium. So it causes sodium to go from your kidney tubes to your blood. So sodium to go from your kidney tubes to the blood. So it's called reabsorption. So reabsorption, that's when the substance goes from your tubes to your blood. So remember that water is a follower. It will follow the solute wherever it goes. So if that solute of sodium goes to the blood, then what will follow into the bloodstream? Water, right? Water will follow into the bloodstream. Therefore, it will increase the blood pressure. So it will increase the blood pressure. So that aldosterone plays an important role in regulating the fluid and electrolyte balance within the body. So you get a drop in blood pressure, it'll stimulate the production of that aldosterone, which will cause sodium to go from the kidney tubes to the blood. Water will follow, increase in the blood pressure. So that's why they talk about in terms of sodium diets increasing your blood pressure. So sodium, it can cause the retention of water, and then it can cause water to go into your blood vessels and increase the blood pressure. So make sure you know those functions of important functions of lipids within the body. And then those lipoproteins. So those lipoproteins, those cholesterol, which is the cholesterol. So which lipoprotein, the HDL or the LDL, takes fat from the blood vessel and delivers it back to the liver? HDL. Which one is the bad cholesterol, which takes fat from the liver to the blood vessel? LDL. Very good. So now we'll move on to proteins. Proteins are very important structures within the body, which form structural components of the body. Right? They form these important structural components within the body. And the building blocks of proteins are amino acids. So this shows amino acids. Right? They have these amino groups, right? These nitrogen groups which are present on them. So this shows this nitrogen group. So make sure you know that amino acids have these nitrogen groups on them. It's important to know that because we will take those nitrogen groups off of the amino acid and produce urea, right, which is a component of our urine, which is a waste product. So it's a byproduct of amino acid breakdown. So a byproduct of amino acid breakdown is urea, which is a waste product. So when people have protein is a very good and important component of your diet, but if you have too much protein, an excessive amount of protein, it can be damaging because it produce too much of that urea. So this shows examples of these amino acids. So there are 20 different amino acids in total, which make all of the different proteins within your body. So this shows kind of the breakdown of the different structural components of protein. So the primary structure of a protein, that refers to the amino acid sequence. So the arrangement of these amino acids. So view these as little building blocks, which will come together to create these proteins. So this shows these amino acid sequences. So the secondary structure, right, they result from the little hydrogen bonds which can occur between the different amino acids. So by forming bonds between each other, So if you have this sheet of paper, then you can have the hydrogen bonds, which can cause one part to bond to another, to cause folds or little twists to develop. So this shows this alpha helix, right? This little twist that's a result of the hydrogen bonds amongst the different amino acids within a protein. And this shows a beta pleated sheet that is a result of the different hydrogen bonds within them. And then they could then turn into these three-dimensional structures, right? These three-dimensional structures are the tertiary structure. And then if you have two or more of these tertiary structures combined together, that forms a quaternary structure. Think quaternary refers to quattro, that's four, like 4 divided by 2. So two of these globular structures, two of these tertiary structures combined together form the quaternary structure of a protein. So I'll talk about different proteins within the body. So we are what we eat, right? We take the protein from the food that we eat, break it down to its amino acids to create the proteins within our own body. So I wanted to show a brief video which describes how animals take, and so that keratin, that's an important protein that you find on the surface of your skin. So if you look at a rhinoceros horn, it's made up of that hard, tough keratin protein. So that keratin makes up your hair, it makes up your fingernails, and it's this tough, fibrous protein on the surface of your skin, which helps to make your skin water resistant and provide that protective layer of skin. Right? And then also collagen. Right? Collagen, that is an important protein which acts like the glue within the body, helps to connect the different tissues within. It's a very important component of the connective tissue within of the body, found within your skin, within your bones, within your cartilage of your joints. So this shows so cigarette smoke, it can damage the formation of collagen within the skin. So this shows a comparison of twins. Right? This twin didn't smoke, and then this twin did. Then this twin didn't smoke, and this one did. So it shows the effects of the cigarette smoke on the creation of collagen with the skin. Without that collagen, right, it causes the wrinkles as a result of not having that collagen present. So this shows these cells are called fibroblasts within the skin and connective tissue, and they act kind of like spiders, creating a web of that collagen fiber. So these little pizza roll looking structures are fibroblasts, and this little web that create, it is collagen. So you find that within the dermis of your skin, within the cartilage of your joints, within your bones. Very important protein within the body. And then also elastin. Right? Elastin is an important protein which allows your skin to be elastic, allows it to stretch and go back to its original position. And then your muscles, right? Your muscle is made up of protein. So actin and myosin, these are the components of the proteins which play an important role in creating your muscles. So it's always talk about how muscle is made up of protein. This actin and myosin, these are important proteins within the body. So make sure you know the examples of proteins within the body. Collagen within connective tissue, within the dermis of your skin, within your bones, within your cartilage, acts like the glue of the body. Keratin, you find it within your skin, within your hair, your fingernails, right? And that actin and myosin, important muscle proteins. Then proteins also play an important role in the catalysis of chemical reactions. So they make enzymes. Right? Enzymes are made out of protein. So I view enzymes kind of like the construction workers of the body. Right? So you see the hammer, and you see the table that construction worker is created. So if it wasn't for the construction worker hammering that nail into the board, right, that board would never be created, because it would take a long time for the nail to get into that wood. So that the construction worker kind of catalyzes that reaction. He brings the board together with the nail. So that's what those enzymes do within our body. They help to catalyze and help to speed up different chemical reactions. So I talked about that lactase you find within your small intestine, helps to break down that glucose to galactose. So this shows an enzyme, and the substance that the enzyme is acting on, that's the substrate. Right? So for example, the example of lactase. So the lactase would be the enzyme, and then the substrate would be the lactose. So the lactose would go into the enzyme, it would catalyze the reaction, which is the breakdown, and then you get the glucose and galactose. Those would be the products of that reaction that enzyme helped to speed up. So make sure you know, very important, that enzymes are made from protein, because those enzymes, they help to build all the structures within our body, help to facilitate our metabolism, breaking down the different products that we eat. So very important to know. So make sure you know the collagen, keratin, elastin, actin, and myosin. And make sure you know that those enzymes are made out of protein, and also transport. So hemoglobin, that's an important protein on the surface of your red blood cells, which help to transport oxygen back, transport oxygen back, transport oxygen back and forth. And also hormones. Hormones are made out of protein as well. So, for example, insulin, glucagon, other hormones from your pituitary gland are made out of protein as well. And then antibodies. So when you get a vaccination, they take a dead or weakened part of the flu virus. Let's say the flu vaccine. They take, so this would be a cell called a B cell. They take a dead and weakened part of that virus and then introduce it to that B cell in the form of the vaccine. Then that B cell will turn into a plasma cell, which will release these antibodies, these little wire-like structures, which will then provide immunity against the virus or whatever you got vaccinated against. So these antibodies floating within your blood play an important role within your immune system, and they are made out of protein. So when people are deficient in protein, they can have some immune dysfunction because they don't have the building blocks to create those antibodies. And then another important protein is albumin. Right? It plays an important role in regulating fluid balance within the body. So, for example, if you've seen images of children in third world countries who are deficient in protein, there's a disorder called kwashiorkor, where they have those distended bellies. So this would be a child, and this is that distended belly. It's because they are deficient in the, this is abstract, all right. It's because they are deficient in the protein albumin. Albumin, it helps to suck fluid into your blood vessels. So it helps, so we know that water is a follower. It helps to suck fluid into your blood vessels. So if you're deficient in albumin, right, you don't have that protein which helps to bring the fluid into your blood vessels, so all that fluid will leak out. All that fluid will leak out of your blood vessels and will cause edema. So this is that stomach that has edema. All that fluid is within that interstitial environment, right? It's all leaked out, right? The swelling, because they don't have the albumin protein which helps to suck the fluid into their blood vessels, plays an important role in fluid balance. So this shows the action of those enzymes, help to catalyze the chemical reaction. So there's a certain amount of energy that's needed in order to facilitate that chemical reaction. So this shows without an enzyme, this shows with. So it lowers that energy, that called the activation energy, that is needed to stimulate that chemical reaction to occur. So the enzyme helps to decrease the energy needed to facilitate that chemical reaction to occur. This shows the action of the enzyme. So this shows the enzyme. So they have these binding sites where they bind onto the substrate. So let's say this is lactose. Right? That lactose will go into that active site, and then it will break apart into that glucose and glucose and galactose. Or if you're creating protein, these amino acids will go into that active site. The chemical reaction will occur, and then you will have that long chain of protein. So that's very important because certain drugs work on enzymes. So for example, ACE inhibitors, right? That's short for angiotensin converting enzyme inhibitor. So that ACE, it produces a substance called angiotensin 2, which increases the blood pressure, right? By constricting the blood vessels and triggering the release of that aldosterone that we talked about. So by blocking that active site, it stops the production of that angiotensin 2. So it decreases the blood pressure. And they'll talk about that more in A&P2, but just to understand the vital importance of those enzymes. And now we'll move on to talk about nucleic acids, right? We'll talk about these nucleic acids or some other proteins in the body. So melanin, right? Melanin is an example of a protein that we'll talk about. This shows the production of melanin. And if people have deficiencies in melanin, they could get that vitiligo. So melanin is another important protein within the body. So now we'll talk about those nucleic acids. So nucleic acids have important structures which make them up. So think ESPN. Think ESPN. So the E stands for the E in nucleotide. Let's decrease the size of that. Nucleotide. E. So nucleotides are made up of a sugar, a phosphate group, and a nitrogen-containing base. So the ESPN, the nitrogen-containing base, makes up so the sugar, phosphate, and nitrogen-containing base. So we'll take a look at a nucleotide. Right, so this shows DNA. So this shows the sugar, right? That deoxyribose. So that's the sugar. All right, the phosphate. This is the P, the phosphate. And then this shows the nitrogen-containing base, adenine. Right, so those bases, they form the letters which make up your instruction manual. So your DNA, that is the instruction manual to create you. And then within the DNA, they have these bases. Right? A is for adenine, T is for thymine, and we'll talk about the guanine and cytosine. So these are the bases which make up the DNA. So I talked about the sugar and phosphate. So within DNA, you have this double helix. So this is one helix, this is the other helix. So the backbone is made up of the arrangement of those sugars and phosphates. So see, this is a sugar and phosphate linked together. And then the rungs of the ladder, right, they're made up of those bases arranged kind of anti-parallel to each other. So adenine always bonds with thymine. So think AT, guanine always bonds with cytosine. So that's the complementary base pairing that you find within DNA. So if right now that you know that the DNA is the genetic instruction manual, that is made up of the sugar phosphate backbone, it's made up of those four bases, adenine, thymine, guanine, and cytosine. You know now more than the most advanced scientists knew maybe like 60 years ago, right? Because we were just learning so much more about DNA, more so now than we ever have before. So make sure you know those four bases which make up the DNA: A, T, G, C, and those components of a nucleotide, right? The sugar, the phosphate, and the nitrogen-containing base. So the genetic information, and we'll talk about this more in the next chapter, it kind of follows a chain of order. The DNA is located within the nucleus. So view that as the recipe. So DNA is the instruction manual to create proteins. It's the instruction manual to create proteins. So it's the instruction manual to create proteins. So that's within the nucleus. And then to produce that protein, it goes from DNA to RNA, which is another form of genetic information. So that RNA, it is single-stranded. So it is single-stranded. And instead of thymine, it has a base called uracil. Instead of thymine, it has a base called uracil. So think RNA does not speak Thymenglish. It does not speak Thymenglish. It speaks uracil language. Right? It does not speak Thymenglish. So there's no thymine in RNA. And then you go from RNA to create protein. So if you see that melanocyte. So the gene to create that melanin is in the form of DNA, and then they transcribe it in the nucleus, going from DNA to RNA, and then it links together on those ribosomes to create links together to those amino acids to create that protein. So this shows within the nucleus you have, so this little strand, this is DNA. Then that DNA gets transcribed into RNA, so that green ribbon will be RNA. And then those ribosomes are like the chefs, which read the instructions on that RNA to link together the amino acids to create that protein. And we'll talk about this more in the next chapter. So another type of nucleic acid is ATP. ATP, adenosine triphosphate. Adenosine triphosphate. That ATP, it is an example of a nucleic acid as well that is used to, so this shows the structure of ATP. So right here. So it has 3 phosphate groups, and it will transfer that phosphate group to energize those different molecules. So those were the main concepts. So any questions about anything in chapter 2? So we'll move on to chapter 3, which if you're in the lab, we so the cell, that's the smallest unit of living things. So when we go to the laboratory, we'll take a look at some different cells under the microscope. So the genetic information, that DNA is located within the nucleus, and then that little walnut in the center is the nucleolus. So let's say you want to create a protein. You go from DNA to RNA to protein. So did anybody remember the name of the process of going from DNA to RNA? Transcription. So you created that RNA, and then you bring it out to those little small structures where you create proteins. So what's the name of the structures where you create proteins? Ribosome. So studied along this, so along here, what's the name of the structure right here? The rough endoplasmic reticulum. You have studied along that, you have those ribosomes. That is where you create your proteins. That's where you create those proteins. So now you created them. Now you need to package those proteins and make sure they're ready to go. So where's the name of that? What's the name of that structure where you package the proteins and then have them exit out through those different vacuoles? Golgi apparatus. So that Golgi body. So this shows that packaged protein exiting out of that Golgi body. And then the power stations of the cell where you produce that ATP energy, those are the mitochondria. So those mitochondria, that is where you produce that energy. And then right here, so what's the name of function of the smooth endoplasmic reticulum? Remember, lipid metabolism. So the smooth endoplasmic reticulum plays an important role in the metabolism, and plays an important role in the metabolism of lipids. So I wanted to show a video which will kind of help you visualize the different. So we talked about how the outside of the cell is made up of that phospholipid bilayer, double layer of lipids. So all along the surface of that lipid bilayer, you have proteins. Right? Proteins line the surface of that phospholipid bilayer. Right, so this shows that double layer of lipids. Then on the surface you have these membrane proteins which serve a wide variety of functions. So this shows a transport protein which allows substances to get into the cell. This shows a receptor. It's another type of protein that works for cell signaling. So for example, hormones and neurotransmitters, they need to provide signals to the cell. So they bind onto the receptors and then facilitate their response. So for example, certain drugs certain type of drugs are called beta blockers. They work by blocking beta receptors on the surface of heart cells, which respond to epinephrine, which make the heart beat faster. So they block that beta receptor so that epinephrine doesn't bind and the heart rate slows down. So that beta receptor is an example of a protein on the surface of the cell, which acts as a receptor. So those proteins, they act for the function of transport, for the function of signaling, so very important structures. So you need to get substances into the cell. So you need to. So I'll talk about the transport into the cell. So diffusion, that refers to the movement of structures from high concentration to low concentration. So diffusion, that's movement from high concentration to low concentration. So for example, when you spray perfume from one side of the room, it will kind of diffuse and move to the other side. Or if you pump this room up full of gas and you opened up the door, which way would the gas go? Outside, because you have a high concentration inside, low concentration inside, low concentration outside. So that's that diffusion. So this applies to our cells as well. So since that cell is made up of a double layer of lipids, certain substances are able to diffuse into the cell. So for example, lipids are able to enter the cell through simple diffusion. And remember, carbon dioxide, it is nonpolar. And then the lipid membrane is nonpolar, so it's able to diffuse right through that lipid membrane. So diffusion, make sure you know diffusion, that's going from high concentration to low concentration. And substances like lipids, oxygen, carbon dioxide can diffuse into the cell. Facilitated diffusion, that's like diffusion, but those are with substances that are nonpolar. I mean, those are with polar substances. So polar substances, they need to enter into the cell via a transport protein. So they need to be facilitated into the cell via a transport protein. So for example, how glucose. So remember how glucose with insulin bonds with the receptor, allows glucose to enter. So glucose, it enters into the cell via facilitated diffusion. So this is facilitated diffusion. That protein channel is facilitating the entrance of glucose into the cell. It goes from a high concentration in the blood to a low concentration into the cell. So that is facilitated diffusion. Then we have type 2 diabetes. That process is dysfunctional because insulin receptors are no longer sensitive. Glucose is not able to get into the cell. And osmosis, that refers to the diffusion of water. So osmosis, that refers to the diffusion of water. So certain cells can react when they are in certain types of solutions. So a hypertonic solution, so when they talk about tonic, it refers to the amount of solutes in a solution. It refers to the amount of solutes within a solution. So a hypertonic solution, that has a high concentration of solutes. So it has a high concentration of solutes. And remember, water is a follower. It will follow the solute wherever it goes. So if you have a solution, so let's say it is salt water. So the cell, and it's in a hypertonic solution. It has a lot of salt within that solution. So remember that water is a follower. It will follow the solute. So where would the water go in this situation? Into the cell or outside the cell? Out. Remember, it will go out because the water will follow the solute. So remember, the water will follow the solute. So this is an example of a hypertonic solution. So think hyper gets smaller. Think hyper gets smaller. In a hypertonic solution, the cell will get smaller. It will shrink because the water will rush out. And then a hypotonic solution, that is when you have a low concentration of solutes within the solution. So say you got 2 within the solution, then you got a lot within the cell. So remember, since water is a follower, it will follow the solute. Which way will the water go? Into the cell or outside the cell? Into the cell. So it will cause that cell to swell or even explode. So think hypo, it blows up. Think hypo, it blows up because the water will rush into the cell, causing it to enlarge. So think hyper, in a hypertonic solution, the cell will get smaller. In hypo, the cell would blow up. So, for example, if somebody is dehydrated, they'd give them a hypotonic solution so that water will rush into their cells so they'll become rehydrated. But if, let's say, they have like edema or swelling, they'd give them a hypertonic saline solution, which will suck in all of that fluid and make the cells get smaller by sucking in all of that fluid. So think hyper makes the cells get smaller, hypo will blow up. An isotonic solution, that means you have the same amount of solutes in the cell as you have outside. So you have no net movement. So you got 1, 1, this is iso. It will stay the same. So make sure you know if it says, what would happen to a cell in the hypertonic solution, it would shrink. What happened to a cell in the hypotonic solution, it would swell. Isotonic, it will stay the same. So diffusion, that's movement from high concentration to low concentration. But sometimes you need to get substances out from low to high. From low to high. So in that situation, you need to use a process called active transport. Active transport. That is transport that requires ATP to take a substance from a low concentration to a high concentration. So for example, sodium. Sodium is in a low concentration within the cell and a high concentration outside the cell. So in order to get that sodium outside the cell, you need to pump it out using a pump that uses ATP. This process is called primary active transport. Primary active transport. That is when you use ATP to take a substance from a high concentration to a low concentration. So this is primary active transport. And that main example of primary active transport is that sodium-potassium pump, the sodium-potassium pump. What it does, it pumps 3 sodium outside of the cell and brings in 2 potassium. So think 3Na2K. Think 3Na2K. It will pump out 3 sodium and bring in 2 potassium using ATP energy. And remember, you need ATP to take that sodium from the low concentration into the cell to the high concentration now. So this shows that sodium-potassium pump. So this shows inside of the cell, and this shows out. So that phosphate will bind and energize that pump, causing the sodium to exit. And then in exchange, it'll bring in 2 potassium. So it's like an exchange. If you give me 3 sodium, I'll give you 2 potassium. So it's like a hostage exchange. So 3 Na out, 2 K in. So make sure you know that the sodium-potassium pump is an example of primary active transport. Right? Pumping out that sodium from low concentration to high. I think it's like swimming against the stream. It's like swimming against the stream. So in the laboratory, we talked about that process of mitosis. The process of mitosis of cell division. So you need to divide those cells. For example, when you have injured tissues, we need to create more cells. So for example, within your skin, you have this bottom layers of cells called basal cells, which multiply constantly, which undergo that mitosis to make more cells. And then within your blood, right, those stem cells undergo mitosis to create more blood cells. So this process occurs within growth and regeneration. So before the cell divides, it needs to copy its DNA before it splits. So this process occurs via a tightly regulated cycle called the cell cycle. So the cell cycle, the cycle which regulates the copying of the DNA and then the multiplication of those cells. And it occurs in these different phases. So if you're going to make more of something, you need to grow to accommodate the more that's something that you are holding. So the first phase of that cell cycle is called the G1 phase. That's the growth. That's when that cell is going to grow to accommodate that DNA. That's that double DNA. So the first phase of that cell cycle is G1. That's the growth phase. Then the next phase is the S phase. So that S phase refers to synthesis. that is when you copy the DNA. That's when you copy your DNA. So this shows that strand of DNA, and it's being copied. So they're making copies of that DNA. And in the G2 phase of the cell cycle, right, that is when the cell grows once again. It grows once again. So now you have that enlarged cell that has double that DNA. So next you have to, right, split that cell. Then you need to undergo that process of mitosis. So then you have to undergo that process of mitosis. Split that nucleus so you have two cells. So the G1 phase, that's growth. S phase, that's synthesis. You're copying those strands of DNA. G2, you further grow, and then the M phase, that's when the cell goes through that mitosis. So that's that process. It is tightly regulated. So you have these repressor proteins, these which prevent the cell from multiplying out of control. And when those cells multiply out of control, they turn into cancerous cells. So this shows, right, these cancer cells multiply out of control. So normally we have these checkpoint proteins which help to suppress the uncontrolled cell division. So P53, this is an important tumor suppressor gene. It encodes for a protein which will see a cancer cell and kill it. So it will suppress tumors. And if that is mutated, then cancer will develop. So if that P53 is mutated, then cancer will develop. So that's an example of an important tumor suppressor gene, that P53. So once again, I'll go over those stages of mitosis. Those stages of mitosis, remember that, the IPMAT. Think, I passed my anatomy test. So the interphase, so what occurs during interphase? Anybody? DNA doubles. And then in prophase, what's one important event that happens in prophase? Anybody? Go ahead. Yes, very good. The nuclear envelope will deteriorate in the prophase. And then in M phase, right, those chromosomes will align across the middle. And then what occurs, anybody, raise your hand if you give what occurs during anaphase. Remember those chromosomes, they split. Right, those chromosomes, they split. So if somebody's antagonistic, you want to get away from them. So in anaphase, those chromosomes, they split apart in two. And then telophase, that nuclear envelope, it reappears. Then cytokinesis, and that's when the plasma membrane splits and you have those two cells. So make sure you know what occurs in those steps of mitosis. So that tightly regulated process. So this gives a simplification of the gene expression. So the DNA, right? That is the instruction manual to create those proteins. So if you want to express those genes, right? You go take that information from DNA to RNA, and the RNA will go out onto those ribosomes, which will link together the amino acids to create that specific protein. So make sure you know the difference between transcription and translation. Those were the important concepts I wanted to emphasize in chapter 3.